All right, I think we'll get started then and let you guys all chit chat more at the end at the reception. There's been lots of exciting talks going on, and it is a huge pleasure for me to introduce our next speaker, Dr. Lila Davachi. I've been a fan of her work for quite some time, and we were just speaking. This is really the first time we've connected in person to have more of a personal conversation about things. But I just have to say, sometimes it's really wonderful to connect in person, not just in Zoom. So I'm very glad to the organizers today for having put this on and seeing all this interaction. So my first order of business is to tell you a bit about Lila's background. She has an impressive background. She did her Bachelor of Arts in Psychology at Barnard College, then went on to do a PhD in neurobiology at Yale. She did a postdoc at MIT, and then was at NYU until 2017 and is now at Columbia University. Her work is impressive, in particular because it brings together many perspectives, really reflecting her background, with the biological and psychological features being highlighted. Her work focuses on how the mechanisms underlie our ability to form, consolidate, and retrieve memories. Her approaches are broad, not just cognitive behavioral, but fMRI, electrophysiology, particularly EEG, fMRI. She is internationally recognized, has won prestigious awards in many influential papers. I'll just sort of looking down at them. There's quite a list. She's published in Science, Nature Neuroscience, Nature Communications, Current Opinions in Neurobiology, Proceedings of the National Academy of Sciences, Journal Neurophysiology, Neuron, Cerebral Cortex, Psych Science, Trends in Cognitive Science, and Hippocampus. She's really hit all of them. And you'll probably hear from that the variety of journals really reflects her ability to integrate from different disparate perspectives. And I think that's really just one of the highlights for me. Her work in particular has covered, and I'm really excited about this part, how time perception in the past, she's looked at this, how time perception and event segmentation allows us to construct our own realities and perspectives. She's offered insights into the structure of the hippocampus and how it facilitates consolidation, mechanistically looking at event replay and whether event replay can happen not just during sleep, but when we're awake as well. She's done some impressive work as well, looking at how do encoding and retrieval processes interact and how a memory cue triggers hippocampal pattern completion. And not only that, she's managed to incorporate even other higher level cognitive functions, in particular looking at emotion, and has some fabulous new insights into why and how threatening information and events get preserved in all their innocuous, or so we think, detail, and how those might change over time. She's collaborated with Elizabeth Phelps, one of the prior speakers here at Brain Day in the recent past. She has some early work on hippocampal cortical activity with Anthony Wagner. She really has covered it all. And so please join me in welcoming Dr. Lila Davachi to Waterloo today.
Wow, thank you. That was very kind and generous introduction. I'll have to watch that on replay later on. Let's get all my gadgets. I apologize in advance. I have an eye patch, as you can see, and the screen is where I can't see it. So I'm going to be turning once in a while whenever I want to point to something. Okay, so today the title of my talk is The Penumbra of Memory, and hopefully by the end of the talk it'll be clear why I'm using this word that I think about a lot. In the early days, when I started thinking about the role of the medial temporal lobe in memory and cognition broadly, the questions that I asked were more along the lines of, how does memory work? What are the processes that happen and unfold in the brain that support successful encoding versus those that do not? Over time and many, many years and lots of papers later, the way that I approach my research has changed. And so now I ask questions like, what is memory for? Taking into account that maybe our assumptions are wrong. So for example, you might simply think that memory is for remembering because that's how we use it. But I think that's probably going to lead us down the wrong track. And I hope this work will sort of illuminate how I'm thinking about it. And I invite your questions at the end. So what is memory for? And like everything, I'm probably getting this a little bit wrong and a little bit right. But so this is how I've been thinking about it. Now, this is not going to be surprising to you given the wonderful talks we've already heard about today. So I was sort of happy for the preempting of this. But we as humans, as organisms, as animals, we need to build models of the world. And so my work has taken this adaptive lens to understanding memory. We need to, we need models in order to teach us about what environments we might encounter, what might happen in those environments. Stimulus, that includes stimulus associations, stimulus response associations, stimulus outcome relationships, contextual information. So I think that's what I'm using the word for model. We need to understand how the world works. And when building new models, so for example, when you're in a new environment, like my trip to Waterloo, these novel experiences require fast one-shot learning. So this is some of the computational demands of episodic memory are unique. But when encountering, and also, but when encountering new relationships that are not explained by or contradict even our old models, they need to be updated. So the work I'm going to present today highlights some of our work that's suggesting that the brain leverages both online and offline reactivation to facilitate these important operations. So the novel episodic learning, as well as the kind of sculpting or updating of prior models. I recently visited Lisbon. Let's just take an example from a trip. I landed in Lisbon. I'd never been there before. And when walking to the hotel, I passed a couple of markets. The top market had jamon and olives and all these things I knew I already loved. I didn't need to update my model of whether those things were delicious. I knew, and I knew I wanted to go get them as soon as possible. However, there also were these beautiful, colorful cans of marinated fish and sardines that Lisbon is known for. And, I think I had a sardine maybe once when I was like 6. Couldn't really remember if I liked it. But here I was left not really understanding how to think about these items. In other words, I needed to build a new model for myself about how I felt about, sardines. I saw you looking nervous over there. Later that night, I was able to have dinner with a colleague. And we, out of my curiosity, maybe my intrinsic motivation, Jackie, ordered sardines. And the outcome was incredible. Finger licking good. They were delicious. So this is an example of a model that's now come full circle, at least through one, sort of repetition. And what memory can do for us is it should be able to, moving forward prospectively, replay or reactivate this representation such that now my new model of sardines is that they're delicious and rewarding. And so I can understand that, and in the future that can guide my behavior. But also what we've shown in our work is that these moments where we learn new information, whether it's contradictory or consistent, might actually lead to retroactive reactivation of these prior experiences. So at the time that I ate that sardine, what we've seen in the brain is that your brain reactivates my trip to the shop when viewing these colorful sardines. And that's all to say that what memory can do is get these representations in the space of the brain at the same time in order to promote changing those models. In other words, memory can transport you through time in this way, but not just to remember, but actually to update information. Okay, so today I'm going to get through 3 sort of lines of work and maybe a 4th if there's time. The first set of experiments I'll share with you our very first attempts to try to look at memory consolidation and replay in humans using fMRI, and we'll ask how do novel episodic experiences survive or strengthen in memory. The second part of the talk I'll share with you the idea that this post-encoding reactivation, sort of giving away the fact that it will be there, and I'll show you how we measure it, is adaptive and it reflects the prioritization of experiences. So it's not simply strengthening, but that the brain is smart, that during the post-encoding time period, the brain is actually sorting through experiences. And finally, memory reactivation we've shown can also influence subsequent perception and memory. Now, this might be adaptive or maladaptive, and I'll share the results with you and you can help me decide which. I think it probably can go either way. So for the first part, how do novel experiences survive in memory? So just to give you a sense, this is sort of a global map or sort of theory about, I think, how the field is thinking about memory consolidation with respect to the brain. I want to bring up some of the neural regions that we talk about. And I put this in the context of time and sort of the life of a single memory in time. This is very simplistic. So at the time of encoding, by definition, that information that you're now prioritizing in your priority map that Jackie talked about is active in attention. And during this encoding period, it's thought that there's a cascade of distributed processing in the brain that occurs distributed and in parallel across the cortex as well as subcortical regions. But at the same time, there's an index of this brain-wide neural activity in a structure called the hippocampus. Right, so this is keeping track of what the rest of the brain is doing. Later on, during retrieval, so this is sort of an online reactivation, it's thought that when presented with a cue in the environment, only one element of that prior experience, this might unlock the hippocampal representation through a process that's been termed pattern completion, which then can reactivate some of the cortical regions that were originally encountered. So there's sort of this encoding retrieval similarity that's reinstated through hippocampal pattern completion. Now, importantly, it's been discovered, and we looked at this sort of time period between encoding and retrieval, where now we call these offline states. You may not necessarily been thinking about that information. You may be resting, you may be doing something else. And it's clear that even during these offline states following encoding, that there are opportunities for the hippocampal representation to strengthen through replay, and I'll talk to you a little bit about how we look at that in our imaging data, but also there's not just local hippocampal opportunities for replay. This is usually happening on sharp wave ripples, which are these high frequency events, but also that there's opportunities for hippocampal cortical communication. So this replay may be coordinated, happening at the same time between the hippocampus and the cortex. And this is thought to ultimately lead to shared cortical connections that may come to represent those events. So this is sort of summing up a classic idea of systems consolidation, but mirroring on top of it some of the neural mechanisms that we think are occurring. Now we've looked at, just to give you some methods, in the early days around 2008, when we started to do this work, we've used many different methods to try to examine replay or reactivation in the human brain using fMRI. I'm just going to show you an example so we can look at trial by trial levels of replay. But let me first say that the design of the experiments are all very similar. There's usually a baseline rest period before you've had an experience, some sort of experience or task, and then also a post-encoding rest period. And this allows us to ask whether there are changes from baseline to post-encoding periods that reflect your recent experience. So this is our definition of replay. You can do that at the individual trial level. This is just a region of interest in the hippocampus where you can extract the activity from each voxel, simply create a vector that describes the task, and you can ask, is that vector present in baseline and is it more present in the post-encoding rest? You can also create a multi-voxel correlation structure where now you look at the correlations between each of those voxels, and you're now asking, what is the structure that's present during the task? And again, do I see that persisting into post-encoding rest more so than it was available during baseline rest? And the one that I'll focus on today is more systems level. So you can simply look at the level of coordinated activity between two regions during a task. And you could ask whether that coordinated activity persists into future rest, reactivates or replays, and is that there more so than in baseline rest. So in all of our studies, we've adopted the same kind of structure of looking at pre versus post encoding, but we use different methods, and I'm happy to answer questions about these other two, which I won't focus on today as much as the functional connectivity measures. So in the very first study to look at this in humans, this was conducted by Ariel Tambini, who was a graduate student at the time. We basically gave people two novel experiences in the scanner. They were in the scanner. They actually did two tasks: an object-based task and a scene-based task. And not surprisingly, the names of the tasks describe what people were doing. So in the object-based task, people were encoding trials where they presented with faces and objects, and the scene-based task, they were encoding faces and scenes, and they were also asked different questions. So we wanted to make the tasks as distinct as possible while people were laying in the magnet, not moving, looking at the visual screen. So the task for the objects and faces was decide how likely or unlikely this person is to be hanging out with that beach ball. And for the scenes, they were asked how happy or unhappy. So the task context actually differed as well. We used these stimuli because we leveraged them because we know that they activate specific areas of visual cortex. Our aim, again, was to look at hippocampal cortical connectivity. So I'm sure you're aware of the fusiform face area, which selectively processes faces, the lateral occipital complex, which really responds to objects more than other stimuli. And then there's the parahippocampal place area, which responds to scenes. So that's why we chose these trials. And here are those regions on an example subject. You can see they're distinctly localized in the brain. Here's the FFA, the lateral occipital complex, and the parahippocampal cortex. I'm going to focus a lot on this region for the data that I present today. Just to show you that during the task itself, we see what we expect. So during the object face task in blue and the scene face task in red, you basically see equivalent activity in FFA. They both contain faces. The object area LO shows greater activity for objects, and the scene task produces greater activity in the PPA because it has scenes. This should not be surprising. This is just building confidence that people were doing the task. What's really different in this task than any other task before it were these baseline rest scans and the post-encoding rest scans. So bookending both of the tasks that people did, we simply asked them to rest for about 8.5 minutes. So close your eyes and rest and think about whatever you want to think about. They did that as soon as they came into the study, and they did it after each of the 2 tasks. Now, for the analysis, I'm going to show you what we could do, and what we did do is, for example, let's look at this is one participant. This is just the bold signal. This is a really simple analysis taken throughout the 8-minute rest period, and you can look at how highly correlated the FFA and the lateral occipital complex are when people just come off the street into the scanner. And you can see that for most subjects, there's a non-zero correlation. That may not be surprising. This might reflect just the level of connectivity that's present generally in these visual areas. But after doing the object face task, you can see visually, even without even looking at the correlation, an increase in their correlated activity that persisted throughout the 8.5 minutes that we've tested them. We've only ever gone out about 10 minutes in these studies, and it does not decline after time. So as long as they're not doing another task, that's an interesting part of the story, you can see this persistent activity. Now, this is only one participant. I'm going to show you the group data on the next slide. Well, maybe on the next slide. So just to remind you, we had the baseline and the rest task after each of the tasks, so we can compare connectivity after both the scene face and the object face task. And I'm going to show you data today looking at connectivity between the hippocampus and a cortical region. Again, we're interested in coordinated hippocampal cortical replay as an index of memory consolidation. So this is the overall group level data. So what I'm showing you here is connectivity or correlation between hippocampus and the lateral occipital complex during baseline rest, after the object face task, and after the scene face task. And what you can see here is that we get a significant increase in connectivity after the object task compared to baseline. That's also greater than what we see after the scene face task. Now, interestingly, it's tempting to think of this as being only tied to the stimuli that were on the screen, but that wasn't the case, and I'm not going to be able to share all the data with you. It turns out that many regions showed an increase after the object face task, but in general, memory was better for the object face pairs later on than they were for the scene face pairs. So while the encoding activity nicely mirrors the stimuli that are on the screen and that you're processing, the post-encoding brain seems more related to what you're actually going to remember. So to look at the correlations in memory, we can ask, do these resting correlations relate to later memory? And so we can do this on a cross-subject basis because we only have one time point for each participant. Well, actually 2, the post-object and the post-scene rest. So what I'm showing you here is memory performance on the stimuli. We tested their memory. I think I failed to tell you that. Associative memory performance on the x-axis, and what's on the y-axis is hippocampal LO connectivity at baseline. So when you're simply measuring their resting connectivity before they've done the task, that does not correlate with memory in these participants. Each dot reflects a single participant. The blue is the object face task, and the green is the scene face memory. But look what happens if you take the same correlation after they've done the task. So the post-encoding hippocampal cortical correlations after the task show a nice positive relationship with later memory for those preceding items. So this isn't just a persistence of brain states or brain patterns that have been set up by the task, but the extent to which those patterns persist into the future rest period predicts how well you're going to remember those preceding representations. So that's how we can link it and call it memory reactivation. If it didn't have a correlation with later memory, it could just be brain states persisting without necessarily augmenting memory for those preceding items. Okay, so now that we had established, that was the first paper showing that in the field, that you can measure experience-dependent changes in functional connectivity that relate to memory. But then we wanted to ask, well, if this is a signature of memory consolidation, it should act in a certain way. It should be augmented, let's say, with reward or with emotion. And we've done a set of studies showing that you can increase connectivity. But we also wanted to understand, well, what does the brain do when it learns a bunch of different things in the same experience? So in this study, Deepu Murthy, it's a very similar design. Sorry, these are all figures taken from the paper, so everyone has their own style of drawing figures. Again, here are the rest periods. There's a baseline rest and a post-learning rest. And in this study, I want to focus you here on the striped area. This is the encoding. And the reason it's striped is because some paired associates were motivated with high reward. If you remember this, you're going to get $1. And other paired associates are motivated with a lower reward. If you remember this, you're going to get 10 cents. So there's not only a single learning task and experience, but mirrored on top of that is, let's call it a priority map for Jackie. There's this idea that some things are high and low reward. And so when you rest after that experience, does the brain reflect that structure of the world, right? Does it augment representations that are high reward versus low reward? That would be adaptive. That would make sense. Again, we also importantly test memory 24 hours later. We use a delay here because reward, and dopamine in particular, is shown to increase long-term memory without necessarily increasing access to memory immediately. So those memory effects with dopamine take time or consolidation to emerge. Here are the stimuli we used. Again, we used faced word associations in this study, or seen word associations. Again, we're leveraging the stimuli because we know what cortical targets are processing this information during learning. And so the green star signaled to participants, you're going to get a lot of money, well, $1 if you remember this, but there are many of them. And the gray star, you get 10 cents if you remember the association. So in memory, what we see is that 24-hour memory test. This is an associative recall. They're shown the words and they have to recall, they're shown the pictures and they have to recall the word that was associated with it. And we get what we expected. So at 24 hours, we get a significant boost in memory for the high reward versus the low reward associations, right? So memory itself as a behavior is adaptive. That's not surprising, but in this paradigm, we actually were able to see that. And what we did in our analysis here was to ask whether hippocampal cortical correlations, which we've already established to be related to memory consolidation, are actually adaptive. So in half of our subjects, the face trials were high reward and the scene trials were low reward. In the other half of the subjects, it was the opposite. The scene trials were high reward and the face trials were low reward. So we could look at these category selective regions of cortex based on whether they were high or low reward, and not just whether they're scenes or faces, right? And what we saw is that, so here's the data, the delta connectivity from post-encoding to baseline, so this is the experience-dependent change in connectivity between the hippocampus and category selective cortex, whether or not it's the face area or the scene area, we're grouping that together, predicted associative memory for those participants the next day. And that's the blue line. So that did so for high reward, but this connectivity here didn't predict memory for the low reward, at least not with category selective cortex. Also, when we look at the VTA, the ventral tegmental area, which Jackie also mentioned in her talk, we see that even though there was no main effect difference between high and low reward, there's no change in connectivity between the VTA and visual cortex after learning. It was the change in connectivity for high reward areas that predicted associative memory. So both anterior hippocampus and the VTA shows connectivity increases with cortical areas that were high reward that predicted their later memory. So this suggests to us that this post-encoding period isn't just about strengthening all the representations that were encountered. But this is a time where the brain can sort through representations, and it doesn't need to be reward necessarily. We think anything that's been prioritized during learning might then be prioritized in consolidation. Okay, so if you think about a little bit of the data that I've just shared with you, it suggests that the encoding states or neural activity that characterizes any time point in your lives, like for example, this talk right now, has the potential to persist into the future, even though you may be resting. We've also shown, by the way, that when you're doing other tasks like math, we can still measure this persistence. So if you ask people to do math problems instead of just resting, we still see the persistence of these connectivity patterns. I'm just going to refer to them as brain states, right? If you think of this slow kind of this activity in the brain that summarizes these slow oscillations in the fMRI signal. So for us, that was sort of interesting because we thought, well, if these are persisting and okay, fine, they're increasing memory. That was our goal. We were very happy with our result. That was the goal of the study. But then it got me thinking, well, if these states are persisting into the future, when are we actually in the present? How do we know that what we're experiencing and perceiving and remembering in the present is not influenced by what we just did? How, where does the line between the past and the present live? So it actually gets to questions of free will and things like that. Why do I do something now? Is it because of something that just happened? And how do you differentiate those things? We feel like we have free will, of course. So in this next study, we wanted to ask whether prior brain states bias the encoding of current information. So instead of looking for their replay, we want to understand whether they change the way that you perceive and encode novel information. So we chose to look at this with emotion. So we used arousing, mostly negative pictures, and we asked, do affective brain states persist and influence novel encoding? So we can basically ask the question after experiencing a bunch of images that are kind of nasty and arousing, and we have the skin conductance levels to show you that people are sweating. After a block of images where you see negative images, now we show people a block of images that are very neutral but controlled for complexity in terms of number of people and objects and images. Does it actually look like that brain state is bleeding forward? And if it is, then does it influence how individuals remember these neutral items that are now being presented to them? So the design was really simple. We just did a cross subjects design where people saw the same exact items, except the order was manipulated. So people either underwent emotional encoding and then neutral encoding, or they saw the neutral images first and then the emotional images. Okay, so the critical area of contrast I'm going to focus you in on isn't the emotional encoding data per se in the brain, but just the neutral images. Do the brains look more similar to each other because they're encoding the same exact scenes, or do these states look very different across participants because of their prior experience? Okay, does that make sense? All right. So just to show you the skin conductance levels as well. So here is just relative skin conductance levels for the emotional blocks in gray, and then the neutral blocks in different colors, denoting whether they happened after the emotional block or before. And you could see that skin conductance levels, as expected, increase during the emotional picture viewing task, and then they actually go down a little bit during a 9-minute rest, and then they sort of reinstate during the neutral task. So emotional arousal looks like it persists into the neutral encoding block. And again, it's significantly greater here for these neutral images than here for neutral images that are cleanly presented before the emotional images. And I just want to make a note here, I don't think I mentioned this, that these blocks last about 22 minutes or something like that, quite long. So we used a number of different analyses in the paper. I'm just going to share one with you to look at brain states. And so the one I'll share with you is the mnemonic brain state analysis. I don't know why I chose that now. It's probably one of the more complicated ones. But we've done different analyses. One is amygdala connectivity with the rest of the brain. One is looking at an anxiety network in the brain. And this is basically color coding each voxel in the brain based on overall activity for later remembered versus later forgotten items. So it's basically the difference between remembered and forgotten, or maybe you've heard of the subsequent memory effect. So what you can do for each participant is you can treat the brain like it's one long vector, and for each part of the vector, you just put a value in there that represents its subsequent memory effect. And you can then ask, how similar is that vector? How similar is the vector for the emotional trials and the neutral trials? And you can look at that based, I think I've lost this, based on order. And so by one hypothesis, you'd expect the neutral trials to look exactly the same because they're the same images that people are viewing, right? Again, if we can look at neural representation in the brain as simply representing the objects that are there, they should look very similar. But instead, what we see is that the neural patterns that are evoked during the emotional task, the emotional brain states, are significantly more similar to the neutral brain states when the neutral brain state follows the emotional brain state, but not when it precedes it. So the way I think about this is if you used a decoder or a classifier, or if you were an alien, you were trying to figure out what people were looking at, here you would make a mistake. You would tell me that people were looking at emotional pictures more often than here, and you would be wrong. Right. So the brain then isn't just reflecting what people are perceiving, but is reflecting something about their prior experience in this task. Now, does this have an influence on behavior? Does this really matter? And this is probably the more striking effect. So what I'm showing you here is overall memory accuracy for emotional items in gray and for neutral items in the red and the blue. And you could see that we managed to entirely get rid of the emotional memory benefit, which is really hard to do, by simply ordering the information in this way. So in other words, neutral items that followed the emotional task were better remembered later on than the neutral items that preceded the emotional task. So not only did the emotional brain state persist into the neutral picture task, but it improved memory for those items. So the benefits of emotional memory encoding, we sort of leveraged those benefits and we applied them in the subsequent 20 minutes to these other pictures, suggesting that the brain states are bleeding over and they're changing the way that you are encoding novel information. This was just a control that the reviewers asked for, where we controlled for position. You might be thinking about that. So when we showed people neutral images and then more neutral images, you get the same level of memory as in the sort of clean neutral condition. Okay, so what I've gone through so far today is sort of summing up a little bit of the data, and there's a lot more that I'm not sharing with you. So please, if you have questions about it, highlighting our lab's work over the last 12 years, trying to understand, can we measure memory reactivation in humans using fMRI? Let's convince ourselves that this is really the signal we think it is by modulating the kinds of learning that people are doing. But what we've really come out on the other end is showing not only can we measure memory reactivation, but it's actually really adaptive. It's really more interesting to study these post-encoding time states to get a sense, maybe it's a better predictor of what people are actually remembering, or a better predictor of what they were prioritizing during learning. Even though we give them a task, we know that people can't equally apply that task to all the trials. So going back to the beginning of the talk, though, so this is about building a model, right? You can build your model. All these processes probably relate to building a model. Memory replay is really important for strengthening those representations and for sorting them out so that you have the structure of the world. But what happens when your nascent model of the world changes? So we've also done some work to look at violating maybe expectations of that were maybe set up, or teaching people new things to look at whether memory reactivation is really critical in forming or updating these models. So the first question was a behavioral study asking whether new learning can alter the trajectory of consolidation of already encountered representations. So here, it's important to pay attention to the different phases. During phase 1, we call preconditioning, people were exposed to novel items, but they came from 2 different categories. They were either animals or tools. So people are just asked to look at these items. They're neutral items, but they do come from 2 categories. And this is kind of boring. You know, they expected that coming into the Davachi lab, or just looking at pictures of objects. Not very engaging. We call this just like a neutral encoding task. Then they go into the next phase. We don't tell them what's going on yet. After the first phase, we attach electrode leads to their wrists. And we tell them that they're going to undergo some learning now, and that they might feel a mild electric shock to some of the stimuli. So they're sort of prepared that they're going to do this. This is sort of classic conditioning phase, Pavlovian conditioning. During the Pavlovian conditioning phase, each participant gets shocked only to animals or tools across participants. So now they have this arousing experience where now they're learning something new. What they're learning here is that some categories of information are more relevant to me, more adaptive for me than other categories of information. Again, every item is trial unique here. They're never seeing the same item twice. The only relationship they have to each other is their category level. And then after the conditioning, we take the leads off, we show them more animals and tools, again, novel exemplars, and then the experiment's over until we ask them to do a memory test. Now, what we see in memory, here's the data from the first experiment in the paper. We fully expected to see this effect. So during learning, we get better memory for CS plus versus CS minus items. So all the items from the shock category are better remembered than items from the unshock category. That's classic emotional memory. That's not surprising. We also saw that bled forward into the subsequent post period, so that maybe attentional mechanisms were at play. So now that they've learned that animals might shock me, maybe they've sort of borrowed that and they're maybe attending more to the shocked category versus the unshocked. What was really surprising and novel, and the reason this got published in Nature, was this effect. We also found that the CS plus items from the preconditioning phase were better remembered later on than the items from the CS minus phase. So remember, these were being encoded and only seen before anyone had learned anything about conditioning. There was no shock, there was no learning. So this effect must have been driven by later learning here. So here, I think of this as that people are able to update their models and sort of rescue these weak memories for subsequent consolidation. So this is just summarizing. We did several other experiments in the paper. What we found here is we get that effect. This is the 24-hour effect. That's this dot. We also get that effect at 6 hours, but we do not get it when we test immediately. So that's how we know that it's dependent on consolidation. You do not see this effect right away. It takes time for it to emerge. And you don't get that effect if phase 1 items are strongly encoded. So if your model is weak and you're available to update it, then it can get updated. But if your model is already strong, these post-learning effects do not modulate that. And that's a critical prediction of tag and capture theory, if you're familiar with tag and capture. And again, for any item that was seen during fear conditioning, you get CS plus minus CS minus greater than CS minus for all time periods. You get that effect right away as well as at a delay. So this is a retroactive memory benefit that's not just about arousal or novelty increasing memory for everything that you have learned. It's selective here, and that's different than what had been shown by James McGaugh and a lot of the like foundational animal work. So the structure of learning here is actually retroactively enhancing some memories over other memories. Now, what is the neural basis of this? I'm sorry if this looks blurry. In a subsequent paper, we adapted this study to now scan it in fMRI to ask what's happening during conditioning. Is there reactivation, and can we measure it? So in order to look for reactivation, we, sorry, I don't have a figure here. I'll go back here. What we did is while people were encoding animals and tools, between each of the trials, we peppered in a series of scene stimuli, and we just used that to use the PPA as a context tag. So that phase 1 learning now is associated with a bunch of scenes that are completely irrelevant, but we know that the PPA is really, really active and is going to tag that learning context. And we did that so that later on we could train a classifier on scene stimuli as well as scrambled scenes. And then we applied that classifier at the time people are getting shocked, actually during the whole phase. So in each trial, during the conditioning phase, we could apply the classifier and say, how much evidence is there here for scene information? There's no scene information present during the classifier. So we can sort of assume that any scene relevant information may be related to the reactivation of the prior learning context. And what you see here, this is seen evidence plotted on the y-axis as a function of early and late in conditioning, and we get early effects and not late, interestingly. It's sort of a detail of the data. But what you can see here is that at moments where people are anticipating shock or getting shock, we see greater evidence for seen information in the brain, suggesting that they're reactivating that prior context. You get that both in the right and the left PPA, and perhaps more convincingly, the extent of reactivation. So seen evidence for early CS plus trials, that's this data point. So if you look at participants, there's some variation along the mean. Participants who show more seen evidence, more reactivation of the prior context, are also those people who show better memory for those individual items. RME refers to the retroactive memory enhancement. So they show better phase 1 memory for CS plus items versus CS minus. So I know it's a lot of detail, but the point here is that during the new learning, the moments of arousal or anticipation are associated with memory reactivation. And we think of this as online because people are doing a task. Your brain is reactivating the past, perhaps in order to make sense of the world, right? To create a new model. And the model should not only include, in this study, the model should not only include the cow here. It's not about the cow. The cow's not shocking you, right? It's about animals in general. So it makes sense to reactivate recently encountered animals, right? So the example, so does that make sense? So if we had taught them individual exemplars, we might not see this effect. We think that what the brain is learning is something about these categories and that it makes sense to reactivate them. We also looked at the post-encoding time period, similar to all the studies I presented at the beginning of the talk. So we can look at connectivity between the hippocampus and category selective cortex. Now they're for CS plus versus CS minus items from the study. And what you can see here is that the delta change, so the post minus pre hippocampal connectivity with CS plus visual cortex, this could be animal area of cortex, across people predicts how well they remember phase 1 animals. So after the learning, after the shock learning, we had a rest period, and you could see that this post-encoding brain was now continuing to sort through that information, and connectivity was increased with parts of the brain where the CS plus category we know are processed. You can look at that with CS minus cortex, no relationship. So it's not that hippocampal cortical activity wholesale was increased after this aversive learning, and that related to memory. It was selective to the CS plus cortex, and this is just showing you the differential if you just take CS plus minus CS minus cortex. So the extent of increased correlation between hippocampus and cortical regions predicted how well you would remember those phase 1 items. Just to share with you a cute little study that's currently being done to look at also the idea that moments at which we are presented with counterfactual information. This is a study by Wangjing Yu. In this study, she uses GIFs, and this is a behavioral way of looking for memory reactivation. We are going to scan this, but during COVID, she came up with a really neat way to look for memory reactivation in behavior. So just bear with me. So during learning, you learn that you're going to meet a bunch of characters. They could be people or animated characters, and they are having a picnic. And so you get to see the picnic blanket here, and the picnic blanket has a visual image on it, an object, and we're using a shape wheel to be able to measure memory for the specific shape in a continuous way, just to give you a sense of what the shapes look like. So in a trial, you would see the picnic blanket, and then you'd have to pick what shape that was. So you'd have to go over here and pick this item. This is just to make sure you're paying attention to this background contextual information. But then the interesting thing is now you learn that Pikachu really doesn't like cherries. All right, so you have a sad face, and we've sort of modulated. So Pikachu does not like cherries, and you learn that, and you either learn that 1 time or 3 times in one of the studies. We've done 2 versions of this. And then after this learning, we call this AB learning. Now we participants were doing AC learning, where now the valence either changes or it stays the same, but the GIFs are always different. So you learned that Pikachu really doesn't like cherries, and then now during AC learning, now Pikachu loves cherries. He's jumping up and down. He's really engaged. So this is the same kind of thing. This is more like an everyday example. I love Pikachu. He is an everyday thing in my house, maybe not for you, but more so than the shock experiment, right? We're trying to think about how does this happen in the real world? So you either learn that you learn that now he loves cherries, or you learn that, I forget the name of this actress, but recently have been watching her on Netflix. But anyway, she likes broccoli, and she still likes broccoli, even though the GIF is different. And after showing people the new AC learning, here's an example, really happy. She immediately, within a second, asked them to remember what the shape of the picnic blanket was. That's our measure of reactivation. And so we can see how close are you to remembering the actual shape. This is a really, really hard task. In the scanner, what we're going to do is try and decode these shapes and look for reactivation, just like we did with the scene study that I showed you. But what she's found, and I don't think I put the data in here because I didn't think I was going to have time to go over this, but what she's found across 2 studies now replicated is that people's memory for the picnic blanket is enhanced when there's a change in the emotion of the character compared to when it's the same. So it's these moments where our nascent model, is contradicted in some way, that the brain is more likely or more veridically going to reactivate that prior experience to make sense of it, right, moving forward. So in sum, there's ample evidence for memory reactivation or replay across hippocampal cortical networks. And I think we are just beginning to appreciate the complexity of these processes. Again, that it's not just for memory strengthening, but that there's something really active and the brain is really restructuring information so that it makes sense, primarily building knowledge structures. So it supports memory persistence and integration with prior knowledge. And memory's penumbra, this is the idea that it goes forward and backwards. There's a prospective reactivation and then a retroactive replay, allows us to make connections across time that allow us to learn where we are, what happens here, and what do we do next. And I just want to take a moment to thank all of my current lab members and past lab members, but these are the people that make me happy every day, and they're the real reason I love to go to work. So, and they do all the hard work. So thanks to them and my funders, and to you for listening.
So there's some time for questions. If anyone has questions, just put your hand up and I'll come run right over.
Thank you so much for that talk. I just had a question about the shock experiment. So I was just wondering how the effect of improved memory during the shock conditions for the pre-stimulus. Do you think you get a larger effect if you introduced an action for the participants where they could prevent the shock from happening, so the memory becomes useful if they're able to sort of move from the shock?
So you're asking during the conditioning phase if people had an option to avoid the shock? Yeah. Well, I definitely think that that's a great manipulation to improve the learning of the structure, so that they're more likely to learn that one category is associated with shock and the other isn't. We measure learning over time. By the way, we don't shock all of the items in a category because that's too many shocks for people. We do like some 30% of trials. So there is some learning they need to do in that moment. So that would improve the learning. The question is, I could imagine it going either way, like because the learning of the structure of phase 2 is improved, according to the story I just told, that should enhance reactivation of phase 1 items for future avoidance. Although the other way to think about it is if they can avoid in the moment, maybe the avoidance itself may lead to not needing to reactivate as much. So I don't know the answer. I think it's a really interesting question. There's only been a few labs and experiments that have looked at this manipulation of agency in moments of conditioning, and it does really improve outcomes in terms of anxiety and things like that, but I don't think they've looked at memory reactivation per se, or even memory for the shock context and experiment. So I think it's an area that needs to be studied. It's a good question.
Oh, okay, thank you. Thanks for the great talk. Wanted to ask a question about memory retention. like procedure, you said that memory retention increases if you either have a rest period before and after, or you have a emotionally arousing event, or like sensory arousing event before that. Is that applicable to everyday life tasks? Say, for example, I'm doing a task that I really want to learn and keep as a memory. Say, for example, I'm learning something or reading something. Is that beneficial for me to take a period of time of rest and then do something emotionally arousal and just do the task?
Yes. So there are some studies. So Della Sala has done a series of behavioral experiments where he either injects rest or no rest after learning, and he sees improvements with rest as a manipulation. Now, rest, interestingly, I get this question a lot about whether you need what counts as rest. You need to be resting, and in terms of curriculum development, you also have to think about that. So you might not need to rest necessarily. It might just be mind wandering. Having a cup of coffee, doing something different. It could, we don't know the answer to that. One of the things we're thinking about is if that brain region isn't actively engaged, then it might be able to replay. It might be in a state where replay can happen. So it could be that moving from like reading in schools and math to gym and art might actually be better than doing like reading, math. Like my son has like Monday morning a morning meeting. It's all the important stuff they do in the morning, all right back next to it. And, it might not be a good idea to do reading and math and social studies all together in 2 hours in the morning. You might want to spread those across the day. So in other words, what counts as rest could be resting to the hippocampal cortical system and not you. So for example, if you really want to remember something, it definitely is the case that, you could just like pinch yourself a little hard, have your friend punch you, and you probably will remember that information better. But I don't want to condone violence here. There actually is an old quote from samurais that I wish I had it for you, that where the samurai said that whenever he wanted to remember something really important, he would take his sword and he would cut himself to make sure he was going to remember it. So that is not something that we would do in everyday life, but certainly arousing moments. There has been a lot of work looking at post-encoding novelty. And there's been some experiments that show in a classroom setting, if you have children learn a new concept, and then they either learn they go and they meet a new teacher and they learn a new music lesson, versus going from that new concept to doing something with a similar teacher, but it's an old lesson, that they got benefits 2 weeks later for the group that went and did something novel afterwards. Now, how much you can stretch that, we don't know. I'm hoping NSF will agree that it's worth studying because it's something that I'm thinking of building into a grant. But post-learning arousal, like in this study, has been shown to have some retroactive memory benefits. We just don't know the boundary conditions yet to know exactly how to apply that in everyday life. But the answer definitely is yes. There is evidence that it can have real world effects. Yeah.
Thank you very much. You sort of commented on this, but so in one of your studies, you went from looking at the conditioning, the effect of it, immediate versus 6-hour versus 24-hour. So do we know for example, that particular experiment, probably if I come back after a month to these people, they don't remember probably anything, or maybe in 6 months, whatever is the boundary. Do we know when it comes down? Like, what is the time scale? Is it relative to the intensity of the shock, or in what sense do we have any understanding of the time scale? And also, is it related or not to this type of the shock? Does it have to be a negative shock? Could it be like, hey, your lottery ticket won? The negativity or positivity of the shock, would it affect or not? Thank you.
Yeah, so to answer your last question first, both reward and shock we use as manipulations of learning the structure of the world, and they both work. They're both motivating. They both increase in dopamine and norepinephrine. They both probably have similar neuromodulatory systems. The reward learning and shock learning are different in terms of the memory representations that you take away with you. So learning to avoid a shock you get really, really good at it. Learning to work towards a reward, you get equally good at. But later on, if you're working towards a reward, you get a more flexible, holistic memory representation. The learning, the representation and the memory itself when you're avoiding shock is very focused. It's more like an item level memory. You know where the door is, but you learn nothing about that environment. So yes, rewards do work for sure in the moment to reactivate. In terms of how far it goes out, there's only a couple of studies. My colleague Liz Phelps brought people back after a year and was able to show that some of them, a significant number of them, still remembered the blue square shock them and not the yellow square. So you can remember those details. We haven't done that in this paradigm where every item is trial unique. So it might depend on the complexity of the information. And I'm sure it does. But remarkably, people do have, they have a strong association with being in that lab and getting shocked and then what happened in the shock. So they do remember it for a long time. We don't usually bring people back after that long to know, to, sort of answer your question about what is the timeline, but it certainly will depend on the amount of arousal. Obviously, we're in a domain of mild electric shock. We're not in the domain of trauma, right? Which you never forget. And also the complexity of the learning. And then, of course, there's the big variance in post-encoding connectivity, we think. So what your brain is doing after, for whatever reason. So I don't have a great answer for you, but I'm agreeing that it probably does have to do with the magnitude. Yeah.
There's an unhealthy persistence of that interaction, leading them to forever or for a long time, longer than normal, have a strengthened association between that negative shock and what was really pretty neutral. But it's interpreted differently because that connectivity is maintained over a longer period of time. And I wondered if anyone had looked at that.
So we are currently doing 2 different studies in collaboration with collaborators at the medical school where we are scanning people with major depressive disorder, some of which have suicidality in the past or thoughts of suicidality and some who haven't. And what we're asking both, we're looking for the emotional hangover effect. So the extent to which emotional experiences persist and influence encoding, I call that the emotional hangover. But we're also showing them different trials of stimuli, some that are happy and some that are negative, so positive and negative stimuli. And we want to see whether their medial temporal lobe hippocampal cortical system, just as you suggested, is replaying more of the negative events versus the positive events. And could that be kind of a neural correlate of rumination and mood, that when you're when you're feeling down, you tend to be more likely to call other experiences when you were down. Now that's all experimental. We're giving them the trials. In another line of work where we're working with patients with delusions, schizophrenia patients with delusions. Now they have one overarching delusion usually that is really persistent, and we're going to do our experimental task. We're doing something a little bit fun and risky, which is we're actually going to scan them while they're reactivating their delusion in the scanner, like as if they're undergoing a CBT session, because we want to understand not really what's happening in the brain when they're recalling their delusion, but we want to look at their brains after rest to see what networks are engaged. And does it look like a stronger level of replay? And how is that related to anxiety, of course? So anxiety is present in all of these domains. So we're starting to look at that with an eye towards whether replay can be maladaptive when it's too strong and too selective.
Thanks. I'm curious about the role of the amygdala in these kind of experiments that involve both context and consolidation. So I know a lot of classical conditioning paradigms will look at, pairing shocks and stimulus of various kinds, as well as looking at how the context that the animal is in might affect the recall or might be, on its own, cause the animal to exhibit a fearful response. And yet the amygdala is a much simpler structure that isn't necessarily able to encode as high dimensional inputs as the cortex might. Just wondering if you have any thoughts on whether there's a difference between amygdala and the cortex in this kind of a context.
Yeah, so I left out some data. So we do look in the studies where we use especially negative like shock and negative images and scenes, we do see amygdala hippocampal connectivity that's strongly predictive of memory. And in the emotional hangover study, one of the brain signatures of the emotional state that we used was amygdala connectivity with the rest of the cortex. So it is the case that probably amygdala processing can influence cortical processing. And we do see that that does look more similar in the neutral items. So the amygdala is doing what we kind of predicted it would do based on other studies, but I still don't have a good handle on what that is and what is the amygdala contributing to to memory reactivation in particular. What we're seeing more are that these neuromodulatory structures in the brainstem that modulate the release of dopamine and norepinephrine are really triggered by these arousing events. And their connectivity with the cortex and with hippocampus are predictive of memory, not the amygdala in this particular situation. So I have to leave that to my colleagues who study the amygdala. But in our studies, amygdala connectivity with the hippocampus does predict the emotional memory benefit that we see in all of the studies. So there definitely is some communication between those two, especially the anterior hippocampus in humans. It really seems to have strong anatomical and functional correlative activity with the amygdala. Yeah.
There's one more question up here.
Hi. So I was really curious about the offline replay. I think you mentioned that like doing math, it would still sort of show the effect of the replay and benefit later. I'm just curious how that like lines up with older like consolidation ideas that, you know, introducing any kind of new information and it disrupts it because it hasn't been laid down. I'm just curious if you think that it's the same kind of consolidation in the replay or if it's something else entirely.
Yeah, so I think the older ideas you're referring to were theories. They weren't data. So I think they're not right. It's not. I think that's probably time to update. Speaking of old models, so I think that many aspects of systems consolidation theory are really bearing out on the work that we are doing, but that all of my colleagues are doing now that they are also looking at the post-encoding brain. There's a lot of people looking at this work now, and highlighting that any kind of new learning would diminish consolidation. I think that hasn't really borne out, especially now that we know there obviously is a special role for sleep, by the way, which I'm not mentioning. The first night of sleep is really, really important. What we're looking at here is awake, sort of quiescent periods after novel learning, which may be kind of a unique kind of experience. We like to think that we're learning something new every day, but actually we're going on autopilot a lot of the time and using our schemas to get through the world. Now, it's not entirely, like, no one's really looked at that to know, does it diminish? What happens in a new context? I will share with you another line of work we have in the lab is looking at event segmentation, so that when you can be in context A where certain rules apply, you learn about that context, and then you shift to another context B, and you can identify the first moment of shifting as a boundary between the two contexts, and we do a lot of that in sequential learning. And what we are seeing is that the arousal and novelty associated with the novel context actually triggers a replay of the prior event. Very similar to the kinds of ideas that I'm shared with you today about these affective moments. So it could be that not that the novelty itself is a trigger to replay. Now that's kind of like my pet theory. I don't have a ton of data on it. We have a few data points, but that's sort of where I think we're going to be looking. But that once you're deep into that context, then the prior context seems very far away now. And then you're probably going to get diminished replay. The question is, after you go through A, B, C, D context, which is much like our days are structured, and you only get rest at the end of the day, how does the brain deal with that, right? Is it only going to pick out the peaks, of what was really relevant, or does it have time to replay both of them? Some rodent work is starting to look at this, like distal replay and proximal replay, replay of new environments versus really familiar environments, and we're starting to sort of weave that into our experimental designs as well. So we don't know yet. But I do think that the active, awake, resting brain is a lot, used active, is a lot more active than we give it credit for. There's a lot going on.
So clearly it's great to have breaks in between talks so we can consolidate. I'll pass it on.
Let's thank Lila one more time.